Welcome back to JRS Learn Life Sciences. In this video I am going to explain respiratory system of prawn. Prawn is an aquatic organism and it carries three sets of respiratory organs. They are bronchostigate, epipodites and gills. Let us start with the bronchostigate. Bronchostigate is the extension of carapace. This carapace is present on the dorsal part of cephalothorax region. The inner surface of this bronchostigate has a high amount of blood capillaries so that it provides a respiratory surface for exchange of gases. Next respiratory organs are epipodites. Three pairs of epipodites are present, and these three pairs of epipodites are present on first maxillipede, second maxillipede and third maxillipede. Coxa of first maxillipede, second maxillipede and third maxillipede has outgrowth. That outgrowths are called epipodites or primitive gills. These epipodites are present inside of gill chamber. This is the gill chamber, and this gill chamber is enclosed between bronchostigate and phloron. The space between the lateral cephalothoracic body wall and bronchostigate is called gill chamber. Inside of this gill chamber, these three pairs of epipodites are present on the three pairs of anterior thoracic legs, such as first maxillipede, second maxillipede and third maxillipede. The next respiratory organs are gills. These gills are also present in the gill chamber. The gill chamber is covered dorsally by the carapace and laterally by the bronchostigate and phloron, and it is not enclosed on the ventral side and anterior and posterior parts. So the anterior part, posterior part and ventral sides of gill chamber is completely open. Anterior part of gill chamber has epipodites and the posterior part of gill chamber has gills. 8 pairs of gills are present in this gill chamber, and these gill chambers are present on each side of cephalothoracic region in between bronchostigate and lateral body wall of cephalothoracic region. Gills are primary respiratory organs of prawn. Let us discuss in detail about gills in prawn. There are 8 pairs present in gill chamber. Gill chamber is present in between bronchostigate and the body wall of cephalothoracic region. Bronchostigate is the extension of carapace. These 8 pairs of gills, they arrange in gill chamber from anterior to posterior direction. The first gill, the size is small compared with the last gill. The size of gill increases from anterior to posterior part. Only 7 gills are visible. The 8th gill is not visible. That 8th gill is present beneath the second gill. Only 7 gills are visible, and these gills are crescent in shape structure. They have a central axis, and this central axis or base provides a structural support for gill plates. Gill plates are present on these base in 2 rows. These gills are further divided into 3 types based on their position in the gill chamber and their attachment to the appendages. The first gill is also called podobranch. The second and 8th gills are called arthrobranchs, and remaining gills such as 3rd, 4th, 5th, 6th and 7th gills are known as fluorobranchs. The first gill is known as podobranch, second and 8th gills are known as arthrobranchs, and 3rd to 7th, that means 5 pairs of gills are known as fluorobranchs. So totally 8 gills are present in a gill chamber. When gill is attached to the coxa of an appendage, it is known as podobranch, and when gill is attached to the arthrodal membrane which connects the leg to the body wall is called arthrobranch. When the gill is attached to the lateral body wall, that is called plurobranch. The first gill is called podobranch, second and eighth gills are called orthobranchs, and third, fourth, fifth, sixth, seventh gills are called plurobranchs. The first gill is attached to the coax of first maxillipede, second gill and eighth gills are attached to the arthrodal membrane of third maxillipede. So podobranch is attached to second maxillipede and orthobranchs are attached to the third maxillipede, and all these five pairs of plurobranchs they are attached to the lateral body wall of thorax. The number and position of these three types of respiratory organs of each gill chamber can be represented in the form of a branchial formula. Two branchial chambers are present, or two gill chambers are present on the lateral sides of cephalothorax. So on both sides of cephalothorax the carapace is present, and this carapace is extended as a branchiostigate, and in between these two, that means the cephalothorax wall and branchiostigate gill chambers are present. In this gill chamber, the number and disposition of the respiratory organs can be represented using this gill formula or branchial formula. So totally 11 respiratory organs are present other than the branchiostigate. The branchiostigate also acts as a respiratory organ. First maxillipede, second maxillipede, third maxillipede. These three maxillipedes have epipodites on their coxa. Three epipodites are present. And second maxillipede also has one gill in its coxa, that is called podobranch. The third maxillipede has two gills attached to its arthrodal membrane and they are called orthobranchs. First maxillipede has one respiratory organ, that is only epipodite. Second maxillipede has podobranch as well as epipodites. The third maxillipede has orthobranchs as well as epipodites. Third maxillipede has three respiratory structures, second maxillipede has two, and first maxillipede has one. So totally the maxillipedes have 6 respiratory structures. Next, walking legs. First walking leg, second walking leg, third walking leg, fourth walking leg, and fifth walking leg. All these 5 walking legs and these 3 maxillipedes they are present inside of gill chamber. Next, there are 5 plurobranchs are present, and these plurobranchs are attached to the lateral walls of the thoracic segments bearing 5 walking legs. So they are attached to the lateral walls of the thoracic segments from which thoracic legs arise. Totally 11 respiratory organs are present in one gill chamber. This branchial formula is only for one gill chamber. There are 2 gill chambers present on either side of cephalothorax of prawn. 6 are present in the maxillipede and 5 are present in walking legs. So totally 11 are present. Other than these, branchiostegates are also acts as respiratory organs. 3 types of respiratory organs present in the prawn, and gills are regarded primary respiratory organs. Epipodites, they are leaf-shaped structures and their membrane has richly supplied with blood capillaries and serves as a respiratory surface to exchange gases. Branchiostegate also has a membrane in its inner surface with a high amount of blood capillaries to serve as a respiratory surface for exchange of respiratory gases. And gills are attached to body wall through gill root. Through this gill root, afferent branchial artery enters into gill and efferent branchial artery leaves from the gill. Ventrally, anteriorly and posteriorly the gill chamber is not covered, and the scapognathet of fifth cephalic appendage makes water flow over on the gills. When water flows over on the gills, the respiratory gases exchange takes place. This is how the gills perform respiration in a prawn. Let us see the blood supply to the gills. Gill is a crescent shaped structure and it has a base or axis, and on this axis these gill plates are present on 2 rows. The transverse section of gill look like this. In transverse section, a median longitudinal channel and 2 lateral longitudinal channels can be seen, and all these 3 channels are present in the gill base. From these lateral longitudinal channels, marginal channels arises. These marginal channels are connected to median longitudinal channel. These marginal channels are present in the gill plates. The lateral longitudinal channels and the median longitudinal channels are present in the gill base, and marginal channels are present in the gill plate. The lateral longitudinal channels are connected by transverse channel, and then these lateral longitudinal channels are connected to median longitudinal channels by marginal channels. Afferent branchial channel enters into gill root, pumps deoxygenated blood to the transverse channels which connects to lateral longitudinal channels. Then these lateral longitudinal channels receives the deoxygenated blood from transverse channels which has got this blood from afferent branchial channels. From this lateral longitudinal channel, the blood pumps to marginal channels. In marginal channels, the exchange of respiratory gases takes place and marginal channels are present in the gill plate. Then the blood from marginal channels is collected by median longitudinal channel. From median longitudinal channel, this blood reaches to heart by afferent branchial channel. From afferent branchial channels, the deoxygenated blood is pumped to transverse channel. From this transverse channel, blood is collected by lateral longitudinal channels, and lateral longitudinal channels pump blood to the marginal channels. In marginal channels, exchange of O2 and CO2 takes place. Then the median longitudinal channel collects this oxygenated blood. From median longitudinal channel, afferent branchial channels collects the blood. From afferent branchial channels, the blood reaches to heart. From heart, all the body organs receives oxygenated blood. Let us see once again the blood supply in the lateral channels and marginal channels and median longitudinal channel. From afferent branchial channel, blood enters into this transverse channel. From transverse channel, blood reaches to these longitudinal channels. From these longitudinal channels, the blood reaches to marginal channels. From marginal channels, blood reaches to this median longitudinal channel. From median longitudinal channel, this blood is collected by afferent branchial channel. From afferent branchial channel, the blood reaches to heart. So in marginal channels, the exchange of respiratory gases takes place. The scapognatite of fifth cephalic appendage makes water flow over these gill plates. When water flows over these gill plates, the respiratory gases exchange takes place. Oxygen from the water is diffused into blood, and carbon dioxide from these marginal channels is diffused into water. So this is how respiration takes place in 8 pairs of gills of prawn. Thank you for watching this video. Please subscribe for my channel to get more updates.